Hi, welcome back anatomy students. Today we're going to talk about how our body acquires our specific immunity. Okay, at this point we've already talked about our first two lines of defense. Our first line being try to keep everything from getting in the first place. Our second line, if it does get in, we're going to treat it the same. We're going to not differentiate between streptococcus and staphylococcus. In the third line of defense, we are going to differentiate between the two. So in order to do that, we have to recognize the difference between a pathogen and an antigen. We also will talk about the different types of immunity. So we'll classify immunity as either being active or passive and being natural or artificial. The B cells and the T cells make up your third line of defense or your immune system proper. So we'll talk about those as well as comparing our primary and secondary responses to those infections. So our third line of defense is fighting off pathogens by recognizing their antigens. So recognize the difference between those two words because they both end in gen, so that makes them very similar and easy to confuse. So gen, as in genesis, is the formation of, right? So pathogen is the formation of disease. The individual organism is the pathogen. So up here we have the Ebola virus. So Streptococcus aureus, Staphylococcus, Ebola, Salmonella, they're all organisms, bacteria, viruses, parasites. That's your pathogen. On the surface of their membranes are protein markers called antigens. Just like on the surface of your red blood cells, we already talked about those. You have antigens. So the antigen will elicit an antibody response. So that's what the name refers to. So the antigens are the portions, the protein markers that are on the membrane surface of the foreign invader. So once we're exposed to an antigen, we start to develop immunity. And we can develop that immunity either actively or passively. So actively, you're actively involved. You make the antibodies against the pathogen. Passively, just that. You're passive. You let it happen to you. So the antibodies are given to you. So on top of active or passive, you can also classify things as artificial or natural. So actively, you make your own antibodies after naturally being exposed to the virus. So the kid next to you has the flu. You, by chance, were exposed, and you now will make antibodies against it. Or artificially, you went to the doctors, you got an immunization shot, you exposed yourself to the flu antigens, and then you actively made antibodies against it. So immunity can be active and natural or active and artificial. So active means you're making the antibodies. Natural means by chance you came across the antigen. Natural, it was on purpose. Artificial, it was on purpose. Passively, so the antibodies are given to you, can happen, again, naturally or artificial. So naturally, by chance, the antibodies from the mother will cross the fetus's placenta, and the baby will get the antibodies from the mother. That also happens through mother's milk. So again, naturally, by chance, the antibodies transferred from the mother to the baby because it moved through the mother's milk. Artificially, you can get an antiserum, which is plasma plus antibodies. So sometimes you have fast-acting diseases and you don't have time to make antibodies yourself. Antibodies take about 2 weeks to make in order to kill off a disease. But if you had a fast-acting disease, I like to refer to Ebola. That's going to be pretty fast. You don't have time to make the antibodies yourself, so you want to get your hands on some antiserum, some plasma that already has antibodies in it. So again, naturally, by chance, the mother passed the antibodies to the baby. Artificially, on purpose, you were administering an antiserum. Maybe it's an antivenom against a toxic, a venomous snake. You don't have time to make antibodies against that chemical, so you're going to get a shot against it. So that was artificial. It was on purpose. So the third line of defense is our immune system proper. So remember, that's a functional system, and it includes our lymphocytes. And we've talked about these ones before. So we have B cells and we have T cells. So both are made in the bone marrow. All your white blood cells are made in the bone marrow. They are going to develop their immunocompetency or their maturation in different locations. That's one big difference. So the B cells will stay in the bone marrow, and they will develop immunocompetency here. And to say immunocompetency, I mean that they gain that identity that allows them to identify foreign invaders. So their receptor sites become specific and unique to recognize a different invader. So that happens in the bone marrow, and they will stay in the liquids of your body, so the humors of your body, so the blood and the lymph. T cells will move out of the bone marrow immature, and they'll move into the thymus gland where they'll receive hormones that advance their maturation, and that's where they'll gain their immunocompetency or their specific shape to recognize a specific invader. So for mnemonics, I like to keep my B's together and my T's together. So the B cells are active in the blood and mature in the bone marrow. The T cells are active in the tissues and mature in the thymus gland. Okay, so I'm talking about specificity, which is what gives your immune system its diversity. So receptor sites are going to identify different individual antigens. So this receptor here could be an antibody. This portion will be the same in all of them. That's unique or universal, but this portion here, the purple portion, that is unique to this individual antibody. So this is where the variation comes into play. This is where the specific shape that matches a specific antigen occurs. So you have lots of different cells, and as you meet new antigens, you create new shapes to match those antigens. So your lymphocytes are circulating in the blood and lymph. They'll come across antigens in the blood, the lymph, or in the lymph nodes, spleen, thymus, tonsils, and Peyer's patches. We've talked about those before. So here you can see specific shapes. This one has kind of a U shape or horseshoe shaped. This one is more V shaped. So this star will not match this receptor here. So that B cell won't recognize it. But this star will match with this receptor and turn this B cell on, and then that B cell will start making copies of itself so that it can fight off that infection. So making copies is called proliferation. So up here you see I have 4 different B cells, each with a different receptor. Only one of these receptors will match this particular antigen. So when it does, it starts making clones of itself. It proliferates. And some of them will be used right now to fight that infection. They make antibodies, and they're called plasma cells. So you can think of plasma cells as antibody factories. The other set that is made at the same time, these clonal cells, are used for future identification. So these are called memory cells, and they'll recognize that antigen in the future. Okay, so talking about those plasma cells and those memory cells brings us to primary and secondary responses. So here is 3 different B cells, right? And they have different receptors, and only one of them matches these antigens. So in this case, it's the one with the red antibodies. Usually they're different physical shapes, not colors, but this one makes copies. Some will be used right now to make antibodies. Those are your plasma cells. Others will be used later to recognize it in the future in order to mount a second response. So over here you have the primary response, which is the first time you've been exposed. I would like this graph better if there were days along the bottom here. So it takes 10 to 14 days to reach the maximum antibodies. So this is the point where your antibodies are outnumbering your antigens, and so they're able to kill it all off, and you can start feeling better. So you notice the first time you're exposed to an illness, you are sick for a couple of weeks before you start feeling better. Now, you have those memory cells which are going to stay in your bloodstream and in your lymph and in your lymph nodes. And if you're exposed to that same antigen in the future, well, within a few days, you've made way more antibodies than you did the first time. So it's, first of all, faster, 3 versus 2 weeks, 3 days versus 2 weeks. And it's more efficient because you create more antibodies in that short period of time. You're able to kill off the antigens at a quicker rate. And it's also protective because it's going to recognize them. Instead of being responsive, it's going to be proactive, and it's going to take care of the pathogen before it gets a chance to wreak havoc on your body. So that's all I have for our survey of the specific immune system. You should be able to compare organism versus protein, so pathogen versus antigen. Active versus passive, whether or not you made the antibodies. Natural versus artificial. Was it by chance, naturally, or on purpose through some laboratory shot? B cells in your bone and blood, T cells in your tissue and thymus. Primary, Takes a while, not as effective. Secondary, you see it again, the memory cells are in play, and you are able to be your own superhero. So, thanks for listening. Oops, I hit